But we're going to go ahead and get started because I don't want to waste anybody's time, and I want to make sure to leave some time for questions about the final and stuff. So game plan for today is take a look at Blackboard, talk about what was in the email that came out on Friday, do a short trip through chapter 14, which is all about parturition, and then end with a time for you guys to ask questions. So I'm going to go ahead and share my screen. We'll look at Blackboard together. There we are. All right, so we are officially in dead week. Not that you guys don't know that. I'm sure you're feeling it like the rest of us are as well. So just a couple of quick announcements to reiterate in case you haven't had a chance to look at the email that was sent on Friday. This week there is new material, but there is not a quiz. So this week, you guys will see videos about chapter 14, which is all about placentation, and a really brief overview of the process of parturition. And again, there's no quiz due this week, so I don't like giving quizzes and stuff during dead week, but the chapter 14 material or the week 15 material will be a part of the final exam. So just keep that in mind. A couple dates for you guys to keep in mind is this Friday, May 13th. So this is first and foremost Pardon Day. So if you did not get an assignment turned in, you can still turn it in for up to 50% of the total points by 11:59 p.m. on the 13th, which is this Friday. In addition to that, if you want to do some extra credit, you can get upwards of 20 points of extra credit by doing 2 peer-reviewed evaluations. And so the specifics about that are over here in the extra credit module. And so that's where the submission link will be as well. The big thing that I ask for you guys on the extra credit is that if you're going to do 2 peer reviews, put them all in the same pile or in the same file. So it's one PDF that you're uploading. It just helps me to keep everything a little bit more condensed and straight. All right, and then finally, just a quick mention about the final exam. So yes, we are having a final exam. It will be contained in the week 16 folder, which will pop up next week, or actually will pop up this week on Friday at noon. And it's going to close on Friday, May 20th at the end of day. So you've got 7 days to work through that final exam. I'm not going to take any late submissions. So it's on you guys to make sure that you start early, that you have good Wi-Fi, and that there's no major issues happening. That's fully on you guys at this point in time. The reason for that is because I have very little time to get everything corrected and grades submitted. That's why I would encourage you to work on the exam earlier in the week rather than later in the week, just so you don't run into any problems. The final exam is going to be a combination of question types. So there's going to be multiple choice, matching, true, false, short essays, ID, anatomy, anatomical identification, those type of things. And so you will have 120 minutes or 2 hours to complete the exam. And so it's not going to shut down at the end of 2 hours and submit on your behalf. You can keep working to answer the questions, but you will see a reduction in points if you go over the 2 hours. So just please keep that in mind. And then I really suggest you guys study. So you've got about 13 to 14 weeks worth of questions that have prepped you for the individual quizzes. I would go back and use those as study guides. And so that's really the big announcements this week. So Pardon Day and extra credit are due this Friday, May 13th by end of day. The final exam folder will open on Friday the 13th and will close on Friday, May 20th by end of day, and needs to be taken in one sitting and is limited to 2 hours or 120 minutes. All right, so also in the week 14 folder, you will see a note guide for placentation and parturition, as well as a short video lecture as well. All right, so with that, any questions about the schedule for this week or due dates? Happy to take those right now. Great. All right. Just one other comment. I'm hoping to get through everybody's semester projects by Friday, hoping to. That way you guys have everything corrected before you go into that final exam. That is my goal. I think I should be able to do that based on how my calendar looks this week. So just hang in there, but you probably won't see those posted until Friday. All right, let's go ahead and switch gears and talk about kind of the end part of reproduction. And so I'm going to share my screen with you and we'll do, there we go. All right, so let me go back really quickly. All right, so through this course, we have really worked through, and I'm hoping you guys are seeing the slideshow. If you're not seeing a slideshow, let me know. But we've worked through everything from a reproductive standpoint, clear from anatomical structure to prenatal development, and we're all the way here at kind of what I think of as the end of the cycle. And we are with parturition and lactation. And so I really like this end of the cycle. It's a lot of fun. I've done a little bit of research on it when I was in graduate school. There's a whole lot about the whole parturition process that we don't know as scientists. So this is definitely an area that is continuing to develop from a research perspective and gather more information. All right, so last week we talked about not only sperm in the female tract, but the process of fertilization and the development of that embryo. And so as that embryo gets bigger, has more cells accumulate, moves down the uterine horn into the uterine body and starts to attach, not only is that attachment portion stopping maternal or indicating maternal recognition of pregnancy, it at that point in time is going to continue to grow into a fetus and attach in the uterus. And so we're going to fast forward to the end of gestation with the beginning of this lecture and look at final prepartum steps, so pre-birthing steps. And so during this time, we will see formation of the placenta occur. So the attachment that is occurring between the fetal side and the maternal side is actually that developing placenta. So that placenta is going to attach to caruncles or endometrial cups, depending on what species we're actually talking about. And those areas are going to be a point of blood exchange, nutrient exchange, and tethering. And so that placenta becomes really important because, and it's really a cool concept because the placenta prior to that embryo being, or that egg being fertilized was not even a part of the anatomical structure. So it's growing from that embryo. And the other cool part is that it is essentially becoming an endocrine organ throughout parturition. So that placenta, as it forms, is going to gain endocrine function, which means that it has the ability to respond to and produce hormones. And so that placenta becomes really important during the initiation of the parturition or the birthing process. And so we first need to kind of go back to chapter 2 really briefly and talk about these uterine differences. So with different types of uteri, we have different types of placenta. So in a sow and a mare that are kind of our monogastric species, they have a diffuse placenta. And so we'll look at what that looks like here in just a second. But that is going to be a very different attachment arrangement compared to dogs that have a zonary placenta, or primates which have a discord placenta, and especially to cows, sheep, and goats which have a cotyledonary placenta. And so these placentae really look quite different. And what the difference is how they attach to the uterine wall. And so we can see with a diffuse placenta in the sow, we have, even though we have a diffuse placenta in the sow and the mare both, you can see that they look very different. So we've got kind of the endometrial layer where the chronic villi of the placenta is actually invading the endometrium, and that is how that placenta is attaching. And then we have the mare, which where we have kind of this concept of microcotyledons, but it's on the placenta side. So the placenta here shown in red is actually invading this endometrium and putting out a web of tissue and blood and blood vessels. And so even though both of these species have what we consider a diffuse placenta, it looks very different. Let me just make sure that I'm telling you everything that I need to. Yes, okay, we're good. So when we look at the dog that has a zonary placenta, what that actually means is that placenta is actually attached here kind of in the center of the placenta that is surrounding the fetus. And so this placenta has 3 distinct zones. The first one is the transfer zone. That is the very black part here. And then we have the pigmented zone, which is going to be the part in the middle. And the pigmented zone is really where the local region of maternal hemorrhaging is actually taking place, or necrosis. And so that's why it looks a little bit different here. And then we have the non-vascular zone. And so that non-vascular zone is where we actually see that band of tissue form around the conceptus. And so this is where nutrient transfer actually occurs. And so here's where we've got the nutrient transfer actually happening. So our discoid placenta, like we see in primates and in humans, is very different in the fact that the placenta is only going to attach at one place in the uterus. So for example, this would be like a front placed placenta here. And so what ends up happening is that placenta will invade the endometrial layer, and it actually consists of a round patch of chronic tissue that forms that fetal membrane interface. And so the vessels, the blood vessels that we actually see from the exchanging zone, which is down here in the bottom, this exchange zone, merge and form the umbilical cord, okay, or the umbilici vessels. And that is what supplies blood and nutrients to the fetus. And so the vascularization of the chorion part of this, and the chorion is going to be that centermost part of the fetal membrane, is immersed in pools of blood, and in those pools of blood is where we see actually metabolic exchange take place. And so the placenta in our primates and our humans is going to attach just to one part of the uterus. And we get this exchange of blood and nutrients occurring in a really localized area that ends up being the umbilical cord. And then finally, our cotyledonary placenta that we see in cows and sheep is very different. So it's not like a human placenta at all. So what ends up happening here is we have cotyledons that are going to bind to the caruncles of the maternal contribution to that placenta. So the way I remember this is we have caruncles and we have cotyledons. And one part is associated with the fetus and one part is associated with the uterus or the maternal contribution. And so what those two will actually do is they will actually come together and create a placentome. And so the way that I always remember the fetal or the maternal contribution to that placentome is the caruncle, because the mom always drives the car. So that's a really good way to kind of remember that the maternal side of that placenta is the caruncle. And so even with the cotyledonary placenta here, we have a couple of different ways that attachment can occur. Yes, both of them are occurring through this cotyledon kind of button structure, but the way that the blood vessels and the endometrium is associating with the placenta is different. And so in the cow and the giraffe, we actually have what we call a convex cotyledon, where the you actually see a lot of like many finger-like projections here. And so these villi are originating from the chorionic tissue, and they protrude in towards the lumen of the uterus. And so that's why our arrows are pointed this way. So the caruncle or the uterine contribution is actually invading the placenta or the fetal contribution. The opposite of that is with the sheep and goat, and this is where we actually have a concave relationship, to where the chorionic tissue is pushed inward. And so we are forming a concave interface between the chorion and the maternal contribution. So we actually have blood vessels that are pushing in rather than the uterine contribution, which is pushing out. And so it from the outside looking in, they both look like buttons. These are up here at the top. These are the big placentomes and like a growing fetus. This is where all our blood is being exchanged, where all the nutrients are being exchanged. But just realize that the way that those, the chorionic part of the tissue or the fetal contribution of the placenta is invading the endometrium layer of the uterus is very different depending on species. All right, and so this is just a couple of pictures of what this actually looks like. So again, we have those button-like structures or the caruncles that are on the uterus. And we have the placenta or the chorion part of the fetal contribution that is attached to that caruncle. And so in the U, you can see we've got these blood vessels actually running to and from these caruncles. So that's where we see that exchange taking place. And these are interesting because these are kind of a pink skin-like caruncle. When I was doing some research in graduate school, we actually did uterine biopsies on pregnant ewes, and they had caruncles that came out that were solid black. It was really weird to see, but still very cool. So pigmentation can occur in the uterus as well. All right, so just a couple of things to talk about from a placenta standpoint. So the placenta is really, really important because it's not only providing protection for the fetus to grow, but it's also providing the environment for the fetus to grow. And so the placenta is going to regulate that exchange of materials between the dam and the fetus. And so we see we see this type of exchange process in other tissues as well. So an example of this would be like with simple diffusion, which you guys may have learned about in biology or chemistry, where we actually see an exchange of gases and water. That is something that the placenta can help facilitate. The placenta also facilitates what we call facilitated diffusion, where there's active like binding of receptors and uptake of glucose. And then the placenta also will provide or utilize active transport in the terms of the sodium potassium pumps. So this organ, this endocrine organ that we call placenta, does a lot of really different stuff. It's very diverse, and it regulates a lot of things using these different diffusion mechanisms. So just a couple of comments about glucose. So glucose is really, really important for the growth of the fetus because that is the major energy source of the fetus. And so the glucose is derived from maternal circulation. So any restrictions that we see in the feed that the dam intakes can have long-term effects on the fetus. And so we see this it's a very popular area of research right now. There's a group at the University of Wyoming, a group at the Mississippi State, and a group at North Dakota State that specialize in fetal programming. And this is what they're actually looking at, is limiting glucose or making glucose available to the dam or restricting it, and looking at what that does to the long-term effects of the individual. In addition to that, I actually have a really good colleague, and we've sent several students to the University of Nebraska in Dustin Yates's lab, who is using like fetal programming and this type of concept in conjunction with heat stress. And they are looking at how the muscle tissue and the different mechanisms in the muscle tissues are being affected because of things like glucose restriction or heat stress, which is something that we see pretty often in the South with really, really high temperatures during parturition. So those events can have long-term effects on how that fetus performs. Everything from when it's born to how it grows, to the hormones that it produces later on in life, to how productive it is, to how fertile it is, those type of things. So it's a very, very cool part of research. All right, just one other comment, because there is so much vascularization associated with the placenta. If we think about a cow, let me just go back, or a sheep. We go back and look at all of these plasmatones that are here, that is an exchange point between the fetus and the mother. Because there is so much of that vascularization happening, toxins can easily be passed through the placenta. So that could be like a cow eating a toxic plant, and the components of that toxic plant going through the placenta and affecting the fetus and those type of things. So that's one thing that we actually see, and I'm worried about as a producer right now, is the presence of lupin out on rangeland. So lupin is a really beautiful purple flower. You guys can look it up to make sure that you see it, but it is, if a cow does eat a lot of it around 150 days of gestation. So right now that would be kind of the fall calvers that we're getting bred in November and December. They can actually see that lupin toxin come across the fetus and affect that calf, and they will come out looking weird, and they don't live very long sometimes. So we do see lupin toxicity every once in a while, especially here in cattle and sheep in rangeland. All right, so our placenta, again, is our endocrine organ. So it is facilitating both a fetal endocrine production and the maternal circulation. And so our placental hormones can do a bunch of things. They can stimulate ovarian function. They can help maintain pregnancy. They can influence how fast or slow the fetus grows, can start to stimulate mammary function. So placental lactogen is going to be a big part of that in producing the placenta. And as we get towards the end of parturition or the end of gestation, it can help with parturition as well. And so you'll see just a series of graphs in the lecture. I'm not going to spend too much time on that today of how hormones increase and decrease throughout gestation. So this one is interesting. This is blood progesterone levels, and we can see in the progesterone, which we call the hormone of pregnancy, does stay pretty high in the cow through gestation, but it does circulate. So it doesn't stay consistently high in the cow or in the mare or the ewe like it does in the woman. All right. And then the parturition process is a pretty hormonally intense process. So the big take-home process here is that first, the fetus is actually going to initiate parturition through a really complex biochemical event. And I'm going to show you what that looks like here in just a second. And the key to starting that process is in the fetus, what is actually triggering parturition to begin is the fetal hypothalamo-pituitary adrenal axis. So the hypothalamus is going to produce a hormone that is going to go to the anterior pituitary gland. Anterior pituitary gland is going to produce ACTH. So ACTH is kind of a stress hormone, and ACTH is going to travel in the blood system of the fetus to the adrenal glands, which sit on top of the kidneys. And the adrenal glands are going to start to produce things like cortisol. And so you guys probably right now with the end of the semester and the stress of getting projects done and studying for finals, you're probably exhibiting quite a bit of cortisol. It's the stress hormone. Okay, and so similarly, these fetus, when they run out of room, basically in the uterus, they get stressed and they produce cortisol. And what that does is it starts to remove the progesterone block on the hypothalamus, and that starts the whole process of myometrial contractions taking place. And so we'll also see, because of the removal of the P4 block, we'll also see increased cervical and vaginal secretions. So when it comes to the stages of parturition, there's three main stages. Stage one is where we actually see the initiation of myometrial contractions. What this looks like is the fetus actually rotating into position for parturition to happen. Stage two is where we see the fetus actually be expulsed from the body. Okay, so this is where the calf hits the ground or the lamb hits the ground. That's the end of stage two. And finally, stage three is where the fetal membranes clear out of the uterus of the dam. And so from a hormonal standpoint, what is actually starting to happen is when that fetus gets stressed, it will remove the progesterone block on the hypothalamus. And just keep in mind that the removal of the progesterone block is basically stopping producing progesterone and having a couple more cleaving events. Specifically, aromatase is going to cleave androstenedione, gosh, it's been a long time since I've had to pronounce that, into estradiol. And that estradiol production is what fully removes that progesterone block. And with those estradiol with the estradiol production, we see myometrial contractions take place. In addition to the myometrial contractions started by E2, the placenta and the endometrial layer of the uterus will start being produced as well. So prostaglandin F2 alpha will be produced to help remove the progesterone block. So PGF2 alpha is going to be produced by the endometrial layer of the uterus. It's going to go to the ovary and it's going to lyse the CL on the ovary. That helps to remove that progesterone block. As that CL regresses, we're going to see decreasing levels of progesterone. And so when we look at all of these hormones taking place at the same time, here on the x-axis, we have day 0 as parturition. You can see fetal cortisol is the highest of them all because that offspring is super stressed in the uterus. It's running out of room to grow. It's running out of room to breathe and those type of things. And so that's when we start seeing fetal cortisol increase, progesterone will start to decrease, we'll start to remove the block, estrogens will begin being produced to help continue to remove the block, and we will have those myometrial contractions start for the expulsion of the fetus. So all of these things that are happening is really stage 1. And so stage 1 can happen clear out to 5 to 10 days before the parturition event can even begin. So from a flow standpoint, this is what is actually happening. So as that fetus is undergoing stress because it's running out of room and it can't breathe, the hypothalamo-pituitary adrenal axis is going to be activated. Specifically, ACTH from the pituitary is going to travel to the adrenal glands on top of the kidney and is going to produce cortisol. And so that fetal cortisol here sets off a mad series of events. So fetal cortisol is where we will see placental progesterone being produced, estradiol being produced, an increase in myometrial contractions, pressure in the uterus, the cervix will become stimulated. We'll see a jump in oxytocin as well to maximize the pressure and to start contractions of that labor process. In addition to that, we will also see estradiol become the primary hormone coming off of the ovary, and that is going to increase the secretions and mucus being produced by the cervix to help with lubrication. In addition, fetal cortisol is going to trigger PGF2 alpha to be produced to help lyse the CL that is on the ovary. And in addition to that, prostaglandin F2 alpha is also going to trigger the hormone relaxin to be produced. And relaxin is a really key hormone at this point in time because it will actually target pelvic ligaments to start stretching and relaxing. So when we look at an animal out in the field that is getting close to calving, one of the things that I always teach is to look at the back end of the animal. If their vulva has relaxed, if the muscles around the pelvis have relaxed, then they're probably getting pretty close to start calving, and that's because relaxin is doing its job. All right, so that is all stage 1. Stage 2 is actually the labor process. This is the part that we hope we don't have to intervene. Sometimes we do, but stage 2 actually ends with the calf or the offspring on the ground. And so some things to just kind of think about here, that expulsion of the fetus is definitely started hormonally, but some other things that will aid in this process is the actual position that animal is in. So there is a big reason why calves and lambs and piglets should start or should come out head and front feet first. And the big reason for that is because if I could get my annotation to work, and it's not going to, is because it creates kind of this arrow effect, and it starts putting a lot of pressure on the cervix, and that pressure will start to dilate the cervix. And so that becomes tactile stimulation that is in that nerve response is going to travel to the spinal cord and be messaged up to the periventricular nucleus and start producing oxytocin from which will be secreted by the posterior pituitary. And oxytocin comes back down and helps with those contractions. And then stage 3 is the occurs after the fetus has been or has exited the body cavity. And it really looks like the expulsion of the fetal membranes or the placenta. And so the placenta is not going to be contained in the animal. It actually needs to come out of the animal. If it's contained in the animal, it becomes what we call a retained placenta, and it can cause a lot of infection. There's a lot of bleeding that can take place, etc. So most animals will expulse that placenta within a few minutes, sometimes after they have a calf or a ewe lamb or something like that. But sometimes that doesn't always happen. So I just wanted to give you guys a couple of ways that you can kind of troubleshoot retained placentas. If a cow is hanging on to her placenta, sometimes that can be a breed effect. So we saw that for a lot of years in the Hereford breed in cattle. Sometimes it can be because they have a poor mineral balance. So selenium can really play into retained placentas. So my best advice is, first and foremost, just give it time. Just walk away, make sure the calf or the baby is up and nursing and doing what it needs to do, and just give that animal some time, and they will eventually, more times than not, relax and drop the placenta. If for some reason they don't, after 12 hours or a day, some things you can do to help get them to expulse that placenta is tie it in a knot, and that'll add some more weight to it. It also will actually help to bring up anything that is dragging on the ground that she's going to step on. We don't want her to do that. You can use sulfa boluses, which down the vaginal cavity, that will actually help to cause vasoconstriction. So the blood vessels will start to shut down. And my best piece of advice is don't pull it out immediately. Just give it a few days. I've seen retained placentas stay in cows for 5 days, and we'll give a shot of antibiotics just to make sure that they aren't harboring any bacteria or anything. All right. So that is kind of a really rundown crash course in parturition and late gestation and parturition. So keep in mind too that also going on at this point in time is placental lactogen is being produced from or being produced by the placenta as well, and that is starting alveolar development and secretion of milk. And so we can see that actually occur really rapidly. You'll get a lot of edema in the udder as they get closer and closer to parturition. So, all right. Questions. So I do have one question about extra credit. Is the article selection on anything we pick, anything within the specifications outlined in the extra credit assignment? So basically, I want you guys to look at technology that in a peer-reviewed publication that has been published within the last 5 years. So there's lots of reproductive technology out there. Awesome. What other questions can I answer for you guys?
When is the final exam being opened? I know
noon on Friday the 13th.
Okay, cool. And it's due by Sunday.
End of day, May 20th. Friday, okay, May 20th.
Cool. Thanks.
Yep, you bet. Awesome. Okay, guys. Well, I'm going to hang out for a few minutes. If you have questions, I'm happy to answer them. I'm going to pause recording. Thank you so very much for being an awesome class. This is definitely the online class that has been the most active. So thanks for participating and being here on Mondays, and good luck to those of you that are graduating. Good luck getting through finals, and I hope to see a bunch of you at graduation.